Good morning, everyone. I just want to thank the organizers for inviting me here to give an overview of what we do at the FDA and the research that our amazing lab has been working on for food safety. So at the Center for Food Safety and Applied Nutrition, we put the F in FDA. Not many people realize that the FDA stands for the Food and Drug Administration. A lot of people think it stands for the Federal Drug Administration. So at CIFSTAN, we put the food in there. We are tasked with safeguarding more than $1.5 trillion worth of food and dietary supplements. And to do this, we ensure that food is properly labeled. We are evaluating any new food ingredient that may come onto the market. And in yellow is the stuff that our lab is really working with. We're targeting those unsafe products. We're investigating causes of foodborne illnesses with our other federal partners, the CDC and the USDA. We're also trying to modernize our methods. Can we detect outbreaks sooner? Can we find these pathogens quicker and more easily? So our food is global. 90% of our seafood in the US is imported. Almost 50% of our produce is imported. So when we eat things, we're eating things from all over the world. So when you get sick, it's really hard to determine what did you eat that made you sick and where did it come from. So this is some examples. You know, if we have a fruit platter, we may be getting the papayas from Mexico. We might be getting the melons from Indiana. All of our food is global. So really, we need to keep that in mind when we're doing outbreak investigations. And when we look at how many people get sick every year, over 48 million people are infected with a foodborne illness every year. Salmonella alone accounts for 1.4 million of those cases and 26,000 hospitalizations each year just in the US. This comes as a high cost for foodborne illness. We're looking at up to $10 billion of healthcare costs in just bacterial illness alone. And that's really important that we need to bring those costs down because we need to save money. We need people to stop getting sick if it's preventable. And to do this, we started the Genome Tracker project in 2010. The FDA decided we need to start sequencing things. We used to do PFGE. PFGE is very outdated. We need something more robust. So we started sequencing. And we started depositing all those isolates into NCBI in a public database. And by doing that, we can look at illnesses that we're getting from patients and saying, can we determine what they ate? Can we get an idea of where that food may have came from? And using the epi data, we can try to link those cases and find the cause of their illness. So at the FDA, like I said, we started with PFGE, then we started sequencing in 2010 using short-read sequencing. And now we're looking for the future. We're doing quasi-metagenomics, where we're sequencing from the enrichments to determine what bacteria are in those environments. We want to start looking at more AMR stuff. Can we determine where those microbial resistance genes are coming from? And now we're moving towards the future. And in the future, what we see is the potential of using long-read sequencing for environmental work. Can we go into these different facilities? Can we go into the growing fields? Can we find that bacteria? Can we sequence it quicker? And can we reduce the illness burden? So at the FDA, we like to close genomes because we use those closed genomes as references for all of our phylogenetic analysis. At the NCBI, we have uploaded all of these genomes that we've closed, and we've closed over 1,000. During the pandemic, we switched gears to start using Oxford Nanopore more than PacBio because it was more efficient, it was quicker, we could get the lab work done, and go back home during the pandemic. And using those reference genomes, we can really look at AMR surveillance, we can look at DNA modifications, and long-term evolution of those genomes. The other thing we're tasked with at the FDA is outreach. So to do that, we want to evaluate the sequencing in-house and then spread it out to our partners so that they can go out there and they're the boots on the ground and they can sequence everything. So this year, we are having our first training for Oxford Nanopore at our annual Genome Tracker meeting. We're going to have 9 of our public health and ag labs come into College Park, and we're going to give them the tools and the knowledge to be able to go back and close their own genomes, be able to do in silico determination of serotyping and AMR genes. And we're really excited about that. The other thing we do is international outreach. I went down to Mexico last August, and our partners at Seneca have a gridiron, and I went and showed them how to do sequencing so that they can close their own genomes. Because like I said, our food is global. So those pathogens are all over the place, and we really need to characterize them. The partnership we have with Mexico is through our food safety partnership. It started with produce back in 2014, and a lot of our produce comes from Mexico. Over a third of it does. So it's really important to create these partnerships with different countries. So having all those genomes, what do we do next? So in 2015, we worked with NCBI to increase the capacity of their AMR Finder tool to include genes for stress tolerance. We wanted to look at biocide resistance, acid resistance, heat resistance, and we were able to find a set of genes for Salmonella, E. coli, and Listeria, and we put that into the AMR Finder, so that if you go online, you can look up your isolates and you can see, does this have the quat genes? Does this have the loci of heat resistance? What does that mean? Which brings us to the future. We have all these sequences, now what? We're still having pathogens persist in environments. So really what we need to do is look at functional genomics. How are these bugs persisting in these different environments, and can we determine ways to mitigate that risk? And currently we're doing this using a few different programs. We're working on creating TRADIS libraries. So we're creating these mutant libraries where we are mutating one essential gene for every gene in that bacteria. And then we can pull those genes together, pull those isolates together, put it through an environmental test of some type, maybe put it into a tomato plant and see what is essential in that bacteria for it to persist or infiltrate the tomato and survive. We're also looking at RNA-seq. We were currently using the Illumina platform, but what we would like to do is be able to do full-length transcriptomics with ONT, because this is really going to give us a more diverse picture of the transcriptome than what we'll see with short reads. We've done some work with methylation patterns. It was really exciting to see the other talk on this, but in our side, what we're looking at methylation for is, does the bacteria create different methylation patterns because of the environment that it's in? So when we go into a low moisture facility, these bacteria are somehow persisting without water in a very dry environment. Are they methylating their genome differently to create some kind of persistence factor? The other thing we really want to do is we want to get that answer quicker. So currently it can take up to 2 weeks to go from a sample to an isolate and sequencing and analyze it. We really need to shorten that because during that 2 weeks, more people could be getting sick. So what we would like to do is target, can we sequence at sample collection, or can we sequence during pre-enrichment? And what I'm going to do is talk about 2 case studies that we have done work on where we did just that. So we're all familiar with some outbreaks of E. coli in leafy greens. We've been striving really hard to figure out, can we help mitigate that? We've been doing environmental studies in the West Coast to look at samples that we collect from water, from sediment, from animals, from the air. And what we're trying to do is look for, is there seasonality in the bacteria we're finding? Is there AMR resistance? And can we figure out why E. coli or other bacteria are persisting in these environments? So one of the studies we did, we wanted to look at direct metagenomic sequencing straight from the sample collection. So we went out to an irrigation canal and we used a dead-end ultrafilter to collect 100 liters of water. We brought it back to the lab and we extracted DNA straight from the backflush from this filter. And we used 3 kits. We did the rapid kit, the field kit, and the ligation kit. And because of the matrix, this irrigation water, we saw that the rapid kit and the field kit did not perform very well. There seems to be some type of inhibitors in this DNA that's coming from this water that was just killing our pores very quickly. But what we could do is when we used the ligation kit and we had those rounds of Ampure B cleanup, we were able to detect some E. coli from that sample. We ended up having 1 read classified as E. coli 0157, which is kind of exciting. So we were saying that, you know, the limited detection for E. coli in this sample was, it was hard to find straight from the sample collection. So then we said, well, let's go back to the pre-enrichment. Pre-enrichment is 24 hours. It's not, it's still quicker. It's not 2 weeks. So what we did is we took negative irrigation water that was negative for E. coli, and we spiked in known amounts of E. coli to figure out what is our limited detection for E. coli in this matrix. And what we found was that the limited detection was 7 to 10 to the third CFU per mil. We were able to find 4 reads as E. coli in the lower end of the the lower end of our spike in, and we wanted then to find out what is our minimum number of reads that we need to assemble the genome. So we know that our LOD was 10 to the third, but when we wanted to assemble the genome, we saw that we could close the genomes down to 10 to the fifth. So with that, we are getting the STEC genes, we are getting the EEA genes, and we're really able to determine what the bacteria is that we're looking for. We're able to find that E. coli 0157. So this is a picture of a dirty matrix. There's lots of background of different microorganisms, but we found what we were looking for using pre-enrichment. Another story is powdered infant formula. So we're all aware back in 2022, there was an issue with powdered infant formula and Chronobacter. Chronobacter is a naturally occurring bacteria, but it can be harmless, or it can be harmful to infants and immune-compromised people. So here what we did is we spiked Chronobacter into powdered infant formula and put it through pre-enrichment, and we extracted DNA at this point to sequence. This one, we were able to use the rapid kit, and it performed just great, and we multiplexed all of the samples and ran them. This was a whole different environment for a matrix. This was very clean. You know, powdered infant formula shouldn't have a high micro background. So what we saw was the Chronobacter almost looked like pure isolates when we were sequencing it. What we were able to do is we were able to close all of those Chronobacter genomes out of that powdered infant formula. And when we did WG-MLST analysis on 27 of the closed genomes from NCBI, the strains that we sequenced clustered with the strains from NCBI. We had 2 that were from the same sequence type, and we were able to differentiate them by 34 alleles. And this was really kind of exciting. So although we can't use this data definitively from the pre-enrichment, we were able to find that bacteria to know that we were going on the right track. So then we would keep working on isolating that bacteria and then sequence it at the end. And all of this work has been spearheaded by Dr. Narhal Gonzalez-Escalona and Dr. Maria Hoffman, who I have to acknowledge and here for all of their hard work.